Hello again, everyone. Welcome back to the structural equation modeling part of our course. And today we will talk about latent variables in the SEM. And here is the outline for this part of the lecture, and we will learn what are the latent variables and why to use them, what are the multi-indicator latent variables, how to feed them, and what is the confirmatory factor analysis. So let's start from what are the latent variables. So the word latent means hidden or hypothetical. And latent variables are those variables that are not observed, but their influence can be summarized through one or more indicators which can be measured. And these indicators are very useful when we want to address some complex or conceptual properties of the system, which are very difficult to quantify or measure directly. And early applications of these latent variables in SEM were focused on the modeling of the general intelligence of human. So this abstract concept, is impossible to measure, but it can be approximated using some scores of different tests. For instance, we still use it when we measure the IQ. And one example here is example of the concept of actual biomass, the true biomass, which we try to measure using some specific methods, using some scales. So we have estimated biomass as a partial information of the true actual biomass. And of course, we cannot 100% measure the true biomass. That's why we have some error associated with our measurement. And here I recall the specification operators in Latent and in path diagram notations, which we can be used to define the latent variables. So the latent variables can be defined using the operator equal sign and single tilde, and it means that it is measured by other indicators. And notification for path diagram is such an ellipse. Also, you can see that error variance, which is circle, and our latent variable have a similar notification. It's because the error variances in some way also can be seen as a hidden, as a latent variable. This is because we do not measure error variance when we observe our variables on a field or in experiment, but we assume that there is error variance associated with our measurements. First, let's consider this following simple example of latent variable eta, and it is here represented by one measured indicator x, which is shown by square box as all observed variables are. And what might be first confusing for you is this direction of the arrow. We would assume that if x represents this eta, then direction should be in other way. But here we see that the arrow comes from the latent variable to the observed variable. And this is because this indicator variable x, it emerges from the underlying concept represented by this latent variable. And now also we have the path loading or the path coefficient called lambda here. And this is a relationship between a latent variable and the observed indicator. And we can say that this is a shared variance between the latent and indicator variables. And what makes a good indicator is a lot of such shared variance. So if we have this lambda closer to 1, then this x is a better indicator for our latent variable. Also, what you can see here is an error. It's a delta, and it's associated with our measured indicator. So this implies that our indicator x is not a perfect measure or perfect indicator of our latent construct eta here. So in other words, we can say that there are presumably other factors influencing the correlation between the observed and latent variables. And now we can put this our path diagram into the equation. So that here is the values of x as a result of the latent variable eta proportional to the lambda, its effect on x, plus the error delta. But with this, we assume that our variables are normally distributed. So the mean of latent variable equals 0 and the standard deviation we have here. And the standard deviation is a square root of variance, as you might remember. Also, what I want to say is that we are missing here is the variance of our latent variable. So the latent variable has variance as it is not one value, but there should be distribution of values across individuals or plots or over time, depending on what are our data. So the same is for our residual errors. We always have the variance. So we can show this also by this double-headed links here, double-headed arrows. So by this we're saying that our eta latent variable has a variance associated with that, and the same is for our residual errors. And now, using the variance-covariance algebra, we can derive that the variance of x is equal to lambda squared variance of our latent variable eta plus variance of delta, our error variance. So this is technical stuff. You don't have to remember this, but it's good to know what is behind the idea of these latent variables. So now, when we know the variance of our x, we know it because it's our measured variable. And using this formula, we can quantify how much variance does the latent variable explain. So here is the total variance, and here we extract the variance of our latent variable. And this is how we calculate it. And now the question arises, what is the scale or the unit of our latent variable? Because this is theoretical concept and it doesn't have any units, and it can be any units possible. For instance, if this is body size measured by body mass, it can be kilograms or grams. And also if it would have other indicators additionally here, like body length, it would be also measured in centimeters. So it can take any possible measures. But in order to perform analysis, we need to get to define these units in order to know the variance, the distribution of our eta. And technically, this estimation is done by analyzing the variance of the indicator and covariance between the indicators if we have many of them. So in this way, we need to standardize the scale of our latent variable to the scale of our indicators. For this, we scale the rho coefficient of the effect of eta on x to 1. And this would mean that 1 unit difference in x shows the 1 unit difference in our eta. And if we know the scale, we can quantify the variances then. And we can say that if the variance of our estimated parameters here of eta and delta of latent variable and error, if they closely resemble the variance of our x, then we can say that we explain the data well. Now, when we solved the problem with the scaling of latent variable, we still have one complication here. And maybe you noticed the complication is that we have only one known parameter, which is variance of our observed variable, and two unknown parameters, the variance of errors and of latent variable. So this means that we cannot find a unique solution for our unknown parameters. So our model is not identified. And you might remember the T rule. So in order to identify our model, we should have our T value, which is the number of parameters we need to estimate, our unknowns. They should be equal or more than T max. And T max is a maximal number of parameters that can be estimated by the structure, by this model. So this is simply a number of variances and covariances from the observed covariance matrix. And we can easily calculate it using our T rule from the previous lectures. So we use this very simple formula where S is our number of known parameters. And we can see from this T rule that our model is not identified. It doesn't match this T rule. And this means that our degrees of freedom are negative. So in order to fit our model, we need to have degrees of freedom equal 0 or at least more than 0. And negative degrees of freedom would not allow us to fit our model. So um, even we have scaled our latent variable, so we define the weights of distribution of our latent variable, but we still have not identified model. So what we do here then? In practice, in order to meet this rule of non-negative degrees of freedom for our latent variable models, in addition to the scaling of latent variable, we need to have at least 3 indicators for a single latent variable. So we need to have 3 of these observed variables. Sometimes we can have only 2 of them, but for this we will have to scale both of our regression coefficients, and I will show you how to do it in next slides. In case if we have models with multiple latent variables and they are correlated among each other, then we can have 2 indicators per latent variable. And our latent variable can be exogenous or endogenous. So it can be predictor itself or it can be response. In case if we have endogenous latent variable, then we have other variables affecting it. And if it's endogenous, then we also gonna have the error variance associated with our latent variable. And this is variance in response to our predictor. And then the model where our latent variable is a part of structural equation like shown here, where it has some predictors, or when it itself is a predictor, then this is called a structural model because it has a directed pathway to latent variable or out of it. And in contrast, if we consider only the latent construct, only the latent variable, then this is a measurement model. And this is important when we gonna learn the confirmatory factor analysis because the difference between these two matters there. Now let's take one more example. So for instance, we have the latent variable body size and the explained variable or the measured variable is body mass. And this is very important concept here because body size is often used in many ecological hypotheses like metabolic theory or Bergman rule, but almost always it's represented as some easy measurable quantity such as body mass rather than the complex multidimensional construct. But there are obviously many aspects to body size, and it might be difficult to quantify it, for instance like shape or volume or relief. And latent variable modeling allows us to better approach such multidimensional or multivariable concepts. And we can say by this that this is really a powerful tool that allows better integrate theory and our observed measures. Also important to mention here is that some care should be taken when we construct latent variables, because just because we call it a latent variable body size, it's not always means it is exactly body size. For example, the latent variable body size is indicated by total abundance here, and it might appear sometimes legitimate to say that high abundance may constrain body size in population under limited resources. But is abundance really an indicator of our body size? Can we go and evaluate some ecological theory based on metabolism or other theories using abundance? I think if you go and sample population abundance, we cannot predict the, let's say, obesity, right? Because abundance is not a body size. So we have to remember that we need to be sure that the latent variable reflects the actual property which we want it reflects. And here I specify the benefits of using the latent variables. So first of all, it allows estimated this complex, multifaceted concepts, this theoretical constructs that we cannot measure directly. And other important thing that it reduces the random error in the latent variable. And if we have random error in dependent variables, we have less precisely measured estimates. And if we have random errors in independent variables, then we have underestimated regression coefficients. So it's quite beneficial to have the latent variables. And the next is the multi-indicator latent variables. And these are latent variables that have many or multiple indicators. Also, indicators within the latent construct may cause each other, so they may affect each other. Another thing is that indicators may covariate for some other reasons than being the part of the latent variable. So for instance, if we think of body size as a latent construct and x1 and x2 to be, let's say, height and foot size of human, then we can say that they can covariate because they are constrained by evolutionary development of these two traits by a common reason or by each other. And here by the fact that larger feet in a human are required to support physically the taller body. This is another way how to look on the multi-indicator latent variable. So we can say here that latent variable eta represents the shared information of observed indicators, which shown here by these overlaps. And the shared variance of observed factors indicates the true concept, the latent variable, while other information here is caused by error variance. And here is the multi-indicator latent variable for the body size, which we have talked about. So obviously there are many aspects of body size that may be difficult to quantify, right? That's why we usually use the body mass. But we can also include body shape, body volume, body relief, body length, and body width. All of them are the traits of a body size measure, and all of them can represent this true concept of body size. And this example very nicely shows the theoretical construct as a latent variable. So biodiversity is a theoretical concept which we try to approximate using different measurements like species number, presence of specific functional groups, number of rare species, dominance of species or evenness of species. Also usually it's used like genetic diversity, taxonomic diversity. So all of these are just indices which really try to approximate what is true diversity is. And the latent variable refers to the fact that even though this variable were not measured directly in the research design, but they are the ultimate goal of the project. Another important example of theoretical construct in ecology is ecosystem services, which is hot topic in ecology right now. So the ecosystem services are those functions of ecosystem that have value for human well-being. And we can take many different indicators that can represent these ecosystem services. So it can be yield of system, for instance, biomass of some plants or yield of meat, or it can be quality of this yield, like for instance, amount of specific nutrients or the ratio of carbon to nitrogen. Also greenhouse emissions, or it can be also other functions like pollination or pest control. And this all can be used to measure these true ecosystem services represented by our study system. Also, repeated measurements can be indicators of our latent variable. For instance, here we have fish density as a latent, and we have samples done in a different time series. So sampling campaigns in April, June, and September. And now we can use all of these sampling campaigns and say that these all observations make the fish density in this pond, let's say. Also, multi-sampling can be a part of our latent variable. For instance, we have plots and the different observers go in that plot and count number of fishes there and their density. And then we can take the data from each observer for the same plots and we can construct them as this latent variable. Also, what we can say here is that in addition to these two previous benefits of latent variables, which you already know, the use of multiple indicators in latent variable allows better accuracy in measurement of relationship because of their shared variances. And now when we use many indicators in latent variables, let's say for body size, then we can say that if you would use body mass simply as one indicator versus body size with many indicators, then we correct for our choice of the measurement. So because we choose body mass as a body size, it doesn't mean that this is true value. Now the next part is fitting latent variables. And as you might remember, in order to fit our latent variable model, we need to meet two rules, which are shown here. The first rule is that our latent variable should be scaled to our observed variables. And here we consider a latent variable model with only two indicators for this variable. And Latent scales our latent variable automatically to the first observed variable, and then it becomes the reference for the other observed indicators. And here you can see that now we can break our latent variable into two equations for our two observed variables, and when it's scaled, our lambda 1 is scaled to 1. You can see here the value 1 instead of lambda. However, we have another rule that is non-negative degrees of freedom. And as you might remember from my previous slides, that when we have two indicator latent variable model, we still have the problem with identification of this model. So the problem is that we have two known parameters and we still have four unknown parameters, even if we already scaled one parameter to equal 1. And why it is not identified? Because it doesn't meet our T rule. So it means that we still have negative degrees of freedom, and that's why we cannot fit this model. And the solution for this was that if we have a single latent variable, as shown here, then we need at least 3 indicators for such latent variable. And this is because when we have 3 indicators as shown here, we have 3 knowns and 6 unknowns here. And then our number of unknowns exactly matches the number of maximum possible parameters estimated from the model. And this means that our degrees of freedom are equal 0. And this allows us, this is minimum requirement to fit the model. There are still some ways that we can implement the two indicator latent variables as shown here. The first solution would be to set our coefficients lambda 1 and lambda 2 to be equal. And this will assume that our variable x1 and x2 have equal weight in assessing our latent variable. And we can do this in R by first defining our coefficient for each variable and then make them equal to each other using double equal sign. And this will solve our problem of identifiability of this latent variable. Another approach is if we have measured errors. So we can measure them experimentally, but unfortunately this is done very rarely in ecological and biological studies. So to measure these errors, we have to do experiment repeatedly. For instance, we go into the same plot or the same study microcosm or the same study system, and we measure our variable x1, but we have to measure it many times, let's say 10 times repeatedly. And then we take correlation between these repeated measurements, and if we take 1 minus that correlation, this would be our measured error, measured error variance. And then we can use this coefficient, which 1 minus correlation, and multiply it on this variable. So we say that variable x1 correlate with itself with this coefficient, and this will fix our error variance to the known value here. And as I mentioned, this done not so frequently, but if somebody does this, it has also other benefits, except that it solve our problem of model identifiability. So it increases the accuracy in estimating relationship between our variables. Also, it increases the explanatory power of measured variables. And the latent variable with only two indicators, as shown on the previous slide, can be used only in that special cases. But usually the rule is that we need at least three indicators, as shown here, in order to identify our latent variable. And when we have such identified construct, latent variable, we say that this is our measurement model. And if we include other variables, as you remember from my previous slides, then we can say that this is a structural model. So latent variable, that is measurement model, is a part of the structural model. And when we have a latent variable in our structural equation model, we have to follow two steps. And first step, it's called confirmatory factor analysis. So we have to confirm the structure of our latent variable. For this, we test a relationship with the indicators which we have in our model with our latent variable. And this test will show us whether our indicators make a good latent variable. And only then, when we justify this latent variable, this measurement model, we can use it as a part of the structural model. And now we will do exercises in R, and I will show you how to do both of these steps. For this, we will use data published here in this publication by Travis and Grace. And data called Travis Data CSV file are on Blackboard. And this project that they did is dedicated to the restoration of the salt marshes in Sabine National Wildlife Refuge Park in Louisiana and USA. And the idea was to transplant plants from other areas, and the aim of this project was to understand how the performance of these transplanted plants are affected by their dissimilarity to the local conditions of these salt marshes. So here you can see the salt marshes during the restoration, and what authors did, they transplanted individuals of the salt marsh plant called Spartina alterniflora, and they measured their performance relative to local populations. And in their case, the performance was measured by the number of different variables, which include here stem density, number of inflorescences, clone diameter, leaf height, and leaf width. So they considered performance as a latent variable. And then they wanted to know how the genetic distance of these plants affect their performance. So our task for this exercise is to build this structural equation model with latent variable called performance. But before going into the structural model, we have to do confirmatory factor analysis. So for this, we have to analyze our measurement model, only our latent construct and the indicators. And by this, we have to examine whether our structure of latent variable and all this indicator make a good latent variable. So to do this, we always need first to look at the correlation structure of our data, of our indicators of latent variables. Because if there is a common latent factor like performance here, then the observed indicators, these observed variables, should consistently correlate with each other. So for this, we use a function core to our indicators, to the columns in the dataset that show these indicators. And what you can see here is that they do highly correlate. So the correlation range from around 0.6 to 0.96. And this means that they all really can represent this latent variable. However, if you look closer, you also will notice that there is one excessive correlation between leaf height and width. And this potentially might suggest the influence on these two variables by some other factors not accounted here. So now that we have qualitatively assessed the validity of the latent model, we can fit it and examine the output. And as you remember, we use a sign, equal sign and single tilde to define our latent variable. So I did this here, and also we can use the SEM function to fit our latent variable. But in the Lavaan, you also can use the SFA, like confirmatory factor analysis function, and it will do the same for us. And now you will get a common warning which encountered for this type of models. It shows that some estimated variants are negatives. So this may mean the scaling problem, or this may mean that we have negative error variances estimated. But let's first look on the output of our model, and then we will decide what is the problem there. So the summary of output shows that we have very poor fit here. So the question, what is the problem? We have to explore why is this problem using the modification indices. And as you remember, we can use the function mod indices to our fitted model and get the indices. And as you remember, these modification indices here, they show the expected decrease in the model chi-square. So the larger number would lead to a better fit if this path would be included in the model. And we can see here that there is a strong modification index for the leaf height and leaf width. So this is what we saw also from the correlation. So that this modification indices also suggest to include the correlation between these two traits. And this might presumably come from the common constraints, evolutionary, how these two traits developed. So it can be that the leaves of Spartina have evolved having some limited variety of shapes that it can take, and not from the plant's performance itself. So that's why we can introduce this correlation into the model and to refit the model to see whether this change our fit indices. So here is the path diagram where I showed this correlation. And let's take it into our script. So you see here that I added the correlation shown by this double tilde with leaf height and leaf width. And here you can see the output, the header of the summary, and you can see that our chi-statistics highly improved. So introducing this correlated error has now reduced the chi-square, and we have now acceptably low level to achieve an adequate model fit. So our P is high enough. And this means that we failed to reject our latent construct of plant performance, and now we can use it to evaluate our structural model. But also, you can see here the latent variable estimates. So here are our regression coefficients, and also note that the first loading here has been restricted by Lavaan to 1. So it's default in Lavaan, as I told you previously. And this is for the purpose of identifiability of the model. And now, if you recall what was the original intention of the authors, it was to explore how the native versus non-native genotypes of Spartina influence the performance. And they quantified this as a genetic distance of transplanted Spartina individuals from the local population. So now we added this genetic distance as an exogenous variable, and the performance of plants now serve as an endogenous variable here. So let's test this hypothesis and let's fit the following model. So here is the syntax for this model, where performance is a latent variable with our indicators. Also, we have performance affected by genetic distance now, and we have the correlation between leaf height and leaf width. So this our correlation between indicators of latent variable. So now we fit the model, and let's check the results. And our results show that the model fits the data well. as we can see here from our chi-statistic. Also, we will look into the regression coefficients, which we have for all our latent variables, for the correlation between indicators. Also, we can see that the genetic distance strongly affected the performance, but also strongly negatively affected the performance. This means that the more distant the genotypes of the local population from which the transplants were taken, the worse they performed. Now also we can plot it. So we use the same function from library Latent plot, and we plot our model. And here you can see that it will automatically recognize where is the latent variable. And here you have all our regression coefficients. And now it's time for your task. And for this task, we will use data called body size data CSV file on Blackboard. So the project is on the effect of human activities on macroinvertebrate body size in ponds. So we have a human impact intensity, which affects our ponds, and in these ponds we have the macroinvertebrates where the body size construct need to be created, the latent variable body size. And this body size is characterized by few measures taken for this macroinvertebrates from these ponds. So we have body mass, body volume, body length, and body width. And here is the hypothetical model which you need to use in your first and second steps. So the idea behind is that we have the latent construct body size, which is indicated by four measured indicators: body volume, body mass, body width, and body length. And now we have the human impact as exogenous variable which affects this body size. So the task is to perform the confirmatory factor analysis for the latent variable body size. Then to use the results from the step 1 and to perform the structural modeling by adding human impact variable in it. And then fill in the standardized coefficients and the R-square for the model and the fit measures for the model. And most importantly, think about how to interpret these results. And this was the last task for today and the last lecture. Looking forward to see you during the seminars.